Hello, and thank you for joining us for this webinar. Today, we're going to discuss the Klotho gene and protein. And this is brought to you by Klotho Skin. I'm Gail Humble. I'm CEO of Klotho Skin. I am a geneticist and have a regenerative aesthetic practice in San Francisco and LA. Klotho Skin has been a 25-year journey and something I'm very passionate about. By the end of this talk, I hope you will be as well. The learning objectives include discovering the significance of the age-suppressing Klotho gene and the Klotho protein, to gain insight in their function on a cellular level in vivo as well as in vitro, and to understand how it can actually ameliorate oxidative stress, which actually prolongs cellular life in all organs. We will understand then how this translates into the delay of age-related diseases and why this gene, protein, and hormone is so important in the field of longevity. We will review the latest in vitro and in vivo studies and review all ongoing research by all companies. Finally, you'll learn how to increase your own natural Klotho level. All right, let's get started. What's the problem? Well, we've got an aging population. Starting in 2030, all baby boomers will be older than 65, and Americans will make up almost 25% of the entire population. The number of 85-year-olds will triple, and the country will have 500,000 centurions. 50 million people have Alzheimer's today. By 2050, this will triple to 150 million. So what's the solution? So often in Western medicine, we wait until there's a problem to solve it. But what if there's something that can actually delay age-related diseases? Well, it turns out there may be. We can upregulate the expression of the Klotho gene and increase the production of the Klotho protein. So what is the Klotho protein? Klotho is considered to be the first documented age suppressor gene in all mammals. We're going to talk in a second here a little bit about the discovery, but what's important is it delays aging when overexpressed and accelerates aging when it is disrupted. An overexpression of Klotho can extend lifespan and improve overall function of all systems. Conversely, a decrease in the Klotho gene and production of the Klotho protein is linked to accelerated aging and various age-related syndromes. We know as humans, as we age, our DNA production changes, and there's DNA malformations or incomplete replications. This starts leading to various age-related diseases. We're going to show you in our cellular studies how bathing in the Klotho protein can actually decrease DNA damage by 40%. So what is Klotho? We talked about the fact that the Klotho gene is the first documented age suppressor or longevity gene in mammals, and that it can delay aging when overexpressed and accelerate aging when it's disrupted. It was originally identified in 1997 by Kuru, a Japanese scientist who was actually working on the sodium pump. He accidentally downregulated the production of the Klotho gene in the mouse model, and that mouse model died at 5% of his lifespan with early atherosclerosis, pulmonary emphysema, renal disease, muscle atrophy, neuronal degeneration, atherosclerosis, osteoporosis, atrophic skin, and pulmonary emphysema. So then he took the same mouse model and he upregulated production of the Klotho gene. Now, the average lifespan of a mouse model is about 20 months. This mouse model with an upregulation of the Klotho gene actually lived 31% longer. 31% longer. It's a result of the Klotho protein, which is formed from the Klotho gene, actually increasing FOXO1. FOXO1 is known to increase superoxide dismutase and catalase, and this protects all cells from oxidative stress. Different than an antioxidant, I always like to use the analogy of a bull in a china shop. An antioxidant lets the bull in, free radicals form, damage occurs, the bull gets roped, but some damage occurred. With the Klotho protein, the bull never gets into the china shop. Now, we know oxidative stress is 1/3 of the reasons we age on a cellular level. So it's no surprise the mouse model lived 31% longer. Here we see the famous mice. The one on the left is a normal mouse. The one in the middle is a Klotho deficient mouse. And the one all the way to the right is a 2-month-old Klotho overexpressing mouse. Clearly, he looks younger, stays fitter, gets all the girls, and ages more slowly. So again, the Klotho gene not only prolongs life, but it prolongs the time until age-related diseases occur. This is a close-up of the mice. And I want to interject here. We do know animal studies. This was done by Kura in 1997. So the Klotho gene was named after Klotho with a C. She was one of the three fates. She spun the thread of immortality and was renowned for her wisdom and her beauty. It was changed to Klotho with a K because Kura wanted to give his stamp on some way and recognition of his work. Let's talk about all the things that the Klotho gene, protein, and hormone do. First, we know we covered the fact that it blocks oxidative stress by increasing FOXO1 and blocking all free radical formation. That would be enough. But it turns out it's also co-receptor for FGF23, which is instrumental in phosphate and vitamin D metabolism. It attenuates insulin and IGF-1 receptor activity, which is a key component to metabolic regulation and modulation of lifespan. Perhaps you remember the study done years ago. There were two groups. One group was given all the food they wanted. The other group was basically starved but got their nutrition. Which group lived longer? And the answer is the group that was partially starved but got the nutrition. This is a result of interacting on the insulin pathway, and we're going to see how that works in a second on a cellular level. But Klotho protein does more. It modulates glomerular integrity and supports kidney function. It regulates mineral metabolism. It does not cross the blood-brain barrier because the Klotho protein is a very large protein. It's 120 to 130 Daltons. That's huge. And yet, it can increase cognition. As a matter of fact, at UCSF, they've completely reversed the deficit with Alzheimer's and Parkinson's by giving a peripheral injection of the Klotho protein. And it does this by not crossing the blood-brain barrier. Now, how is that possible? Well, it turns out the Klotho protein does something more. It activates platelet factors, in this case, platelet factor 4, which stimulates new neurotrophic function. It mitigates the risks associated with cardiovascular disease by improving endothelial function. And the one slide we left out here is actually increases nitric oxide production, which, as you know, is important again for all oxygen delivery to all organ systems and cellular levels. So here we're showing what's actually happening on a cellular level with Klotho. When your Klotho protein level is increased, and remember your Klotho gene is making your Klotho protein, You have an increase of something called superoxide dismutase and catalase. This blocks all oxidative stress and results in cellular longevity. So all your cells are living longer. What else is happening is actually it's decreasing DNA damage by 40%. So not only are your cells living longer, but they're replicating more accurately. You're staying younger longer on a cellular level. Here we see how it's interacting on the insulin pathway, which we talked about earlier. You can see Klotho is blocking the autophosphorylation of IGF, activating FOXO, which then increases superoxide dismutase and catalase and blocks oxidative stress. What's interesting is the Klotho level is so important for well-being. It's actually an independent predictor of morbidity and mortality. Older people of the same age, taken next to each other, one with a higher Klotho level, actually has a decreased risk from cardiovascular disease, cancer, and death for all reasons. As a matter of fact, Klotho levels actually correlate to morbidity and mortality. Now, this is a U-shaped curve, and we're not going to spend too much time talking about the upper levels, but at the right concentration, Klotho levels can actually decrease morbidity and mortality. There's an age-related change in Klotho that occurs. Your Klotho level is 6 times the level in your cord blood as the day that you're born. This then decreases by 50% by the time you're 40 years old. And interestingly enough, there's also a diurnal variation. Guess when it hits the lowest point? 3 p.m. That's right, the afternoon sleepies, where there's a 40% decrease in your Klotho level. There is, interestingly, a heterozygote in the human population that actually is 1 in 5, and these lucky people have an increase in Klotho of 15 to 20% naturally. So the Klotho protein actually attenuates and regulates metabolic and modulation factors that increase lifespan on a cellular level. As a matter of fact, in all disease states, your Klotho level is actually decreased. Let's go over them. Generalized aging, which going forward, we have to start thinking about as a disease of sorts, if we can prevent it. Renal conditions, cardiovascular conditions, pulmonary disease, metabolic or endocrine diseases, cancer. Both levels are awfully almost unmeasurable in people with cancer, Parkinson's, Alzheimer's, psychiatric disorders. Now, this is interesting. They did a study, 2 groups of women. One group had children that were born on the spectrum, and they measured the Klotho level and the 2 levels of the mothers. The mothers that had children on the spectrum had a much decreased Klotho level as a result of the bottom one, chronic stress. Autoimmune disease also decreases your Klotho level. So what can we do to change this? We know the Klotho level is incredibly important and a predictor of morbidity and mortality. Is there a way that we can upregulate the Klotho gene and get the Klotho protein in humans? Well, what we did was we started with adult mesenchymal stem cells, which are fat cells banked in a petri dish. We formed a Klotho construct and used a vector, and we're going to explain more about that in a minute. But basically, we were able to upregulate production of the Klotho gene in the stem cells in the petri dish. Now, it's interesting. Stem cells, and I want you to visualize this, sit in a petri dish, and they're alive. They need to be fed an agar and they excrete a fluid. That fluid is called the cell conditioned medium. In that cell conditioned medium, it's reflective of the health and the genetic disposition of the stem cell that's excreting it. By increasing the Klotho gene, we were able to get the Klotho protein and second generation growth factors in the cell conditioned medium. Now, you've heard about cell conditioned medium in certain other products, but this is an older technology. You may have also heard about growth factors as far as PRP. Growth factors that are second generation are quantitatively and qualitatively greater. So here's how we did it. We formed an open reading frame. This is a construct. Think of this as an on or off switch. And then we used a vector, which is like an arrow. And we shot that arrow into the DNA of the stem cell in the petri dish. As a result, we turned on the Klotho production of the stem cell. Now, stem cells are like teenagers. They either want to grow up and differentiate into soft tissue, bone, whatever they're going to turn into, or they want to kick out the genetic mutation that we've done, or they die. So to keep a stem cell alive in an undifferentiated state and be able to replicate it took about 20 years. We finally did it. We took an adult mesenchymal stem cell from fat and we were able to replicate it. Because one stem cell has a very small amount of cell conditioned medium, and we needed a large amount of this to have scalability. After we had the replication of the stem cells, we collected the cell conditioned medium, and we did an ELISA assay to monitor the amount of Klotho protein and growth factors in the cell conditioned medium. The results were impressive. And this is a patented process. This graph shows the increase in the Klotho protein in the cell conditioned medium. We have a few minutes here. We're going to go into the cellular studies. All these cellular studies were done with the cell conditioned medium. That makes up 25% of the face serum, 15% of the eye serum, 10% of the night cream, and 50% of our Klotho complex, which is used for microneedling. In the first of our cellular studies, dermal fibroblasts were taken, and this is in vitro. We had a control, and we had the other group of fibroblasts that were bathed in the cell conditioned medium with the Klotho protein and second generation growth factors. We then used UVA irradiation and looked at the results. This was control versus treated cells. The results were amazing. 40% reduction in DNA damage. This was done by Amelia Labs, backed by an NIH grant by Peter Sikora. Actually, he called me and said, I know it can't possibly be reproduced, but I wanted to let you know what we found. Well, he did reproduce it over 15 times on over more than 250,000 cells. As you know, the UVA damage is what causes tumors. And every time there was a 40% reduction in DNA strand damage. So then we went a little further. We took these fibroblasts and we exposed them to UVA radiation only to equal of 8 hours of sunlight. As you can imagine, we thought the results would diminish, but they didn't. We had the same decrease in DNA damage of 40%. This suggests that Klotho protein is protective, preventative, and regenerative. The reduction was not simply attributed to lower endogenous or starting levels of DNA damage, but rather a clear protective response against oxidative DNA damage. We did 2 other cellular studies. The first one, we took different levels of our cell conditioned medium and we monitored other genes that were present. We found at the right concentration, two other genes were increased. These were HTERT and SIRT6. Now, these are really important genes. I know you've all heard about telomeres. They basically act as a cap at the end of a telomere. On a cellular level, your telomeres don't really lengthen. And as they get knocked down and shorter, the cell then dies. Often this leads to a cell that's called a senescent cell or a zombie cell. And an increased load of these can increase your disease level process. We took another study, and this time we bathed the fibroblasts with UVB irradiation. We actually found an increase in collagen level 4 in our fibroblasts that were bathed in the Klotho cell conditioned medium. So again, it increases HTERT and SIRT6. It's preserving telomeric life. It's protecting against UVA by decreasing DNA damage. It's protecting against UVB by increasing collagen number 4. It increases nitric oxide. It decreases transepidermal water loss, and it increases platelet factors. So in the next part of this podcast, we're going to go into the clinical studies and see what happens when you use something containing the cell conditioned medium on live human subjects. Finally, we'll wind up with talking more about the ongoing research of the Klotho protein, and finally tell you how you can increase your Klotho level every day.